Hello, and thank you for joining us for immunotherapy for ovarian cancer. Immunotherapies are drugs that target your body's immune system to help fight cancer. This session will discuss the promise and challenges of this exciting area of research. Dr. Kunle Odunsi is Deputy Director, the Robert Ann and Lou Wallace Endowed Chair in Cancer Immunotherapy, Executive Director of the Center for Immunotherapy, Professor in the Department of Immunology, and Chair and the M. Stephen Piver Professor in the Department of Gynecologic Oncology at Roswell Park Comprehensive Cancer Center. You can view Dr. Odunsi's full bio in the downloadable conference program. Thank you so much for joining us, Dr. Odunsi, and I'll turn it over to you.
Thank you very much for the kind introduction. Today I'm going to be addressing the question of where are we going with immunotherapy for ovarian cancer? These are my conflicts of interest. Immunotherapy has become the backbone of many types of treatments for cancer in general. If you turn on the news at any time, or even if you look at some news magazines, you're going to find immunotherapy in almost every one of these outlets because it is rapidly becoming the backbone, either alone or in combination with other modalities of treatment for various forms of cancer. So the first question is, what is immunotherapy? Immunotherapy is what the immune system is what we use to fight off bacteria, viruses, fungi, or any invading pathogens into our body. There are many types of immune cells that engage in this fight against bacteria or viruses. One of the most important cells is called the Tcell. And when a Tcell engages its target, as shown here, the Tcell delivers molecules that destroy the foreign target, the invading pathogen. So the question is, how can we induce immune responses or enhance immune responses using different approaches against cancer? So the same principle, exactly the same concept of fighting bacteria or viruses can be used against cancer cells, whereby these T cells deliver a kiss of death. And this, in fact, is a targeted answer in cancer therapies. So another way that immunotherapy has been defined is by one of my friend and colleague, Jed Walchuk from MSK, who defines immunotherapy sometimes as treating the immune system so it can treat cancer. And you'll find out in a moment why this definition is important to understand. So why should we harness the immune system against cancer? Vaccination against infectious diseases, as I've indicated, has proven to be cheap, safe, very successful in preventing illness and death. If you look at this table here, it shows the annual morbidity infections by various infectious pathogens. I mean, look at smallpox at the beginning of the 20th century. Look at how many people it affected at that time. And the list goes on and on. Measles, rubella, tetanus. But look at the numbers recently. We have been able to eliminate smallpox completely with a decrease of 100% over the years because of the immune system, because of vaccination against smallpox. So is there any evidence that immunotherapy can potentially benefit ovarian cancer patients? This is a study that we conducted more than 15 years ago, where we looked under the microscope and looked at the tissue specimens from almost 120 ovarian cancer patients at Roswell Park. On the top panel here, you will find those brown dots. Those represent T cells, especially a population of T cells that are called CD8 positive T cells. So the tissue specimen from the patient at the top contains a lot of CD8 T cells, whereas the tissue specimen at the bottom contains very few T cells. We then asked the question, does this correlate with survival of ovarian cancer patients? And as you can see, patients along the red line did a lot better, with a median survival of 55 months compared with patients along the blue line with survival of 26 months, which was very highly significant. And I would like to acknowledge also the work of George Kukos and his group that published similar observations actually before our report, indicating that the presence of T cells within ovarian tumors appear to confer improved survival of these patients. And the question is, how can we leverage this information for patient benefit? So let's go back to immunotherapy in general. What are the benefits of immunotherapy? The major benefit, one of the major benefits is that the immune system is very specific. It has this unique ability to discriminate self from non-self. So when it attacks its target, it's in a very specific way. It can be used against many types of cancers, even those that do not respond well to chemotherapy. There's potential for reduced side effects commonly associated with chemotherapy. And one of the major benefits of the immune system is that it can induce long-term remissions. So I think we're really just even at the tipping point of cancer immunotherapy, because there has been several FDA approvals for many solid tumors over the last few years, for cancers of the lungs, bladder, breast, melanomas, over the last few years. Look at what happens with traditional therapies. If you look at this curve, patients initially respond, but eventually the traditional therapies fail to work. This curve goes down to almost 0%. What have we done over the last 50 years or so? We combined chemotherapies, we combine with radiation, and we shift the curve to the right a little bit, but ultimately the tumor comes back. We extend lives a little bit, but often the responses are not durable, and few patients ultimately are saved. So here's the potential of immunotherapy. It can shift this curve even further to the right, and in fact, it can even shift it upwards, whereby when you combine different types of immunotherapies or combine with traditional therapies, now you begin to get great durable benefit from immunotherapy. So the question becomes, what are the types of immunotherapy? Because people talk about immunotherapy as if there's only one type of treatment. There are, in fact, many types of immunotherapies. We already talked about vaccination, just like the same way we vaccinate against the flu virus, we can vaccinate against cancer. That is a form of immunotherapy. Another form of immunotherapy is the use of immune checkpoint inhibitors, adoptive cell therapy, oncolytic viruses. So there are different types of immunotherapies. The word doesn't mean the same for all patients. So the question to be asked by each and every patient, if they are considering immunotherapy, is that what is the most appropriate immunotherapy for me? What is the most appropriate immunotherapy for each patient? And that's an important question because it affects the way we think about, the best and most appropriate treatment for each patient. And the second question is, are there some advanced next generation form of immunotherapies? And I'm going to try and talk about all of this. So vaccination. There are two major ways of vaccination against cancer. One is to vaccinate against specific targets on the cancer. One target is called NY-ESO1. Another potential target on the cancer are called neoantigens. I'll talk a little bit more about NY-ESO1. Another strategy for vaccination is to use a patient's own tumor. So with regards to NY-ESO1, this is a target, a protein found on ovarian cancer cells that is not found in any other normal tissue except the adult male testes. We have tested more than 1,000 ovarian cancer patients, and we find NYSO1 expressed in close to 40% of patients. And when we look under the microscope, it looks something like this that I show on the right. So we have conducted vaccination studies against NYSO1 in ovarian cancer patients. And what I'm showing here is that if you look at patients along the blue line on this graph, where the patient's tumor expressed NYSO1 and they did not receive vaccination, about more than 300 patients, the overall survival was 38 months. When you now take a group of patients who receive NYSO1 vaccination, whose tumors are positive for NYSO1, the overall survival was 75 months. And the patients who are NYSO1 negative lie somewhere in between. This is a retrospective analysis, so it has to be interpreted with caution. But this is giving us a signal that vaccination or generation of immune reaction against a target that is expressed on the tumor can in fact potentially lead to clinical benefit. Obviously, this type of data will need to be confirmed in a randomized phase 3 clinical trial. Another type of vaccination is to use a patient's own tumor, and this is elegantly was elegantly presented by Dr. Lana Kanderlaft in 2019 at the ASCO Symposium, and I believe the paper has now been published. This is work out of University of Pennsylvania, where patients were randomized or were assigned to 3 different cohorts. Cohort number 1, a patient's tumor is resected and used to manufacture a vaccine, whereby the vaccine uses a special type of, another type of immune cell called dendritic cells. As you can see, cohort number 1, they got vaccine alone. Cohort number 2 got vaccination with bevacizumab, Avastin, a drug that is very commonly used in ovarian cancer. And cohort number 3 got the vaccine along with bevacizumab and cyclophosphamide. So what the investigators observed is that patients who got the vaccine along with bevacizumab, along with cyclophosphamide, actually did a lot better compared with patients who got cytoxan, cyclophosphamide, and bevacizumab alone, again pointing to the potential role for immunotherapy. So another group of immunotherapy drugs are called immune checkpoint inhibitors, and I'm sure many of us are familiar with these drugs. Immune checkpoints are brakes on the immune system, acting to stop an ongoing and excessive immune reaction. So think about it. When an immune reaction occurs against a bacteria or virus, at some point, the immune system needs to stop when the offending bacteria has been eliminated. So it's like seeing a stop sign. So tumors have hijacked some of these main mechanisms in order to stop the immune system from acting. So the drugs that have been used to release the brake, so the tumor is putting a brake on the immune system. So immune checkpoint inhibitors actually release this brake, and there are many of them that are available, and they are listed here, including Pembro, nivolumab, Tcell, durvalumab, avelumab, and so on and so forth. So these drugs, many of these drugs have been approved for many of the solid tumors, such as lung, bladder, even breast cancer, but not yet in ovarian. So when we look at results of checkpoint inhibitors in ovarian cancer, what we find if you use the checkpoint inhibitor alone is that the results show very little response rates. Response rates in the range of 5, maybe 10%. But what we also see is that many patients achieve some stabilization of disease, what I write here as SD, stabilization of disease, for a while. So these are examples of some of the initial studies. So we conclude from all of this that although there has been spectacular success of immunotherapy with immune checkpoint inhibitors in several solid tumors that led to FDA approvals, the results in ovarian cancer have been modest. So there has been no FDA approval in ovarian cancer. And so the question is why? And there are several reasons for this, in that ovarian cancer uniquely escapes from immune attack in many different ways. Ovarian cancer can overwhelm the immune response, and a lot of several research groups have shown this. It can also hide from the immune system via several mechanisms. It can subvert the immune system. It can put out what are called immunosuppressive chemokines or cytokines to subvert the immune system. It can shield itself. There are ways by which the tumor can shield itself to exclude immune cells, T cells, from entering into the tumor. It can also defend itself by actively deactivating the T cells as they are trying to attack the tumor. So all of this raise the question, are there opportunities to target some of these aspects of ovarian cancer? Are there opportunities for combination therapies? And the answer to that is yes. So many investigators have tried combining chemotherapy with immune checkpoint inhibitors, immunotherapy, either in the frontline or recurrent setting. One of the recent studies was presented by Dr. Lederman at the SGO meeting, whereby a large number of patients, almost 1,000 patients, were randomized into receiving chemotherapy followed by avelumab, which is one of the immune checkpoint inhibitors, or chemotherapy with avelumab followed by avelumab maintenance, or chemotherapy alone followed by observation. What was disappointing with this trial is that progression-free survival was not improved in the avelumab arms when you compare with control. Again, pointing to the challenge of immunotherapy in ovarian cancer. This is another study that was presented at the SGO in 2019. In this case, this was also another phase 3 trial, large trial, where liposomal doxorubicin or Tcell was combined with avelumab. And again, there was no overall survival or progression-free survival benefit in patients in this group. And there was a possible hint of benefit in patients whose tumors express a biomarker called PD-L1. So in our institution at Roswell Park, Dr. Emecheziros recently completed a phase 2 clinical trial combining pembrolizumab with bevacizumab and low-dose cyclophosphamide or Cytoxan. This was a phase 2 clinical trial, and the objective was to test whether this combination will improve progression-free and even overall survival. So what Dr. Zeros did was to enroll patients who either were platinum sensitive, about 10 of them, and 30 patients that were platinum resistant. And this was an open label study in the recurrent setting. So the observations from this trial are very striking in that there was evidence of response in almost half of the patients, either partial response or complete response, about 47% of the patients. And when you combine with patients who had near response or pretty much stabilization of disease, you see almost 80% response rates. The duration of response for patients with platinum-sensitive disease was about 11.5 months. For patients with platinum-resistant disease was 5.8 months, and 25% of all of the patients had progression-free survival that was more than 12 months. This was a very impressive result. Again, it's a single arm phase 2 trial, and this needs to be tested in a phase 3 trial. But what is important is that the quality of life were not significantly impacted by this treatment, whether it's emotional or social functioning or body image, were not significantly affected by this treatment, which again is a very promising result. So one of the mainstays of therapy recently is the use of PARP inhibitors. So is there potential to combine a PARP inhibitor with an immune checkpoint inhibitor? And there are several ongoing trials testing this question. I would like to highlight only one of those trials that was published by Dr. Konstantinopoulos from the Brigham, actually from Dana-Farber. This was a phase 1 and 2 clinical trial of niraparib in combination with pembro in patients with platinum-resistant ovarian cancer. What these investigators observed is a response rate of 18%, with some patients achieving complete response, progression-free survival of 31%, and 12%, and at 6 months, 31%, at 12 months, 12%, as well as evidence that perhaps patients with BRCA mutation had overall response rate that was significantly higher, 47%, and PFS of 8.3 months. Importantly, almost all kinds of patients responded, regardless of platinum sensitivity, BRCA status, or PD-L1 status. This highlights to us that more effective biomarkers are necessary for this group of patients. So I'm going to switch gears and quickly talk about adoptive T-cell therapies. So these are the next generation types of therapies where we can actually take a patient's own T-cells, take the T-cells and engineer it to become, to have ability to recognize tumor targets. So there are two major ways. One is the use of CAR T-cells, which I'm sure most people are familiar with. This has been very successful in liquid tumors. And the second is the use of T-cell receptor engineered T-cells. So we take these cells, we multiply them, grow them up into large numbers, billions of cells, and give back to patients. I consider these cells as foot soldiers that are likely to go into the tumor site and attack the tumor. Essentially, you are fighting fire with this approach. So how do these cells work? Again, if you think about a patient's tumor, we generate the T cells. If T cells, the normal T cells that have not been manipulated, these have not been engineered, they are not able to recognize the cancer cell. Now, when we engineer the T cell by putting in this TCR, T cell receptor, which is the instrument used by the T cell to recognize cancer, the T cell now goes to the cancer site, it engages the cancer, and again releases that kiss of death. So you can imagine that in this fashion, one might be able to get rid of large, bulky tumor. This has been tested by Dr. Steven Rosenberg at the NCI. This is an adoptive T-cell therapy focusing on NY-ESO1 again. And I'll just show you some of the results that are very promising. These are patients with multiple lung metastases from synovial sarcoma. And if you look at the large bulky tumor, pre-treatment in this patient, the CAT scan on the left, you can see this large bulky tumor that has completely disappeared after treatment with this type of engineered T cells. So there are many studies going on in ovarian cancer testing this approach, and I call the current study first generation T cells. So they can attack the tumor cells, but not able to defend itself against tumor counterattack. So what we are now doing is to build next generation tumor cells. And again, if you compare a foot soldier with a soldier who is in an armored tank, that soldier is able to resist counterattack by the enemy. So it's not just about the size of the enemy. The enemy also has to be equipped to resist counterattack. So ovarian cancer immunotherapy, we have to have the mindset of chess grandmasters, because ovarian cancer is always putting out strategies to try and counter the immune system. Many of them are very well known and have been described by our group and others, including starving the Tcells of amino acids, glucose, oxygen, other forms of nutrition. And so we have to utilize counter strategies to overcome this, so that, and this could require additional engineering of T cells to resist multiple mechanisms by which ovarian cancers try to escape. One of such strategy is the use of oncolytic viruses. These are very unique viruses that are coming into immunotherapy of ovarian cancer. They selectively destroy, attack cancer cells, tumor cells, but not normal tissue. And when they do this, they release antigens into the immune system, eliciting an immune reaction. These T cells are elicited within the tumor microenvironment. So not only are these oncolytic viruses destroying tumor cells, they are also inducing an immune reaction against the tumor that will go on to potentially further destroy and eliminate the tumor. So a very successful example of this, again, is in multiple myeloma. There was a paper published by Dr. Anthony Ribas from UCLA in melanoma, showing that these patients with widely metastatic disease had 62% objective response rates, as well as, and 33% of those responses were complete responses. So we and others are beginning to develop novel oncolytic virus approaches for ovarian cancer in order to take advantage of this. So let me conclude. Where are we going with immunotherapy for ovarian cancer? Immune checkpoint inhibitors are promising, but effectiveness may be limited by multiple immune escape mechanisms. So we need to understand these mechanisms and target them. Effective immunotherapy is going to require large numbers of cancer-specific T cells, which we can generate with vaccination, or the adoptive T cell therapy, engineering T cells to deliver large numbers to the tumor. For every patient that is contemplating immunotherapy, either as standard of care or as part of a clinical trial, the question is, which immunotherapy type or combination is most appropriate for that patient, and are there biomarkers that will predict responsiveness for these patients? This will allow us to put together personalized combination strategies of immunotherapy for ovarian cancer patients. And finally, we really need to pay attention to side effects, because sometimes toxicities occur, and the more we combine agents, immunotherapy alone or with chemotherapy, or combination of two types of immunotherapies, we need to watch out to make sure we minimize toxicities in this setting. Here's my acknowledgement slide. I would like to thank all of my collaborators, our clinical collaborators at Roswell Park, members of my team at the Center for Immunotherapy, as well as all of the funding agencies listed here. Thank you very much for your attention.